Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim. I'm Scott Berry, I'm your host, and I am a statistician, as many of you know, Bayesian statistician. And here on In the Interim, we explore things around clinical trial science, medical decision-making, data analytics. This is our space. We have a new studio here where our technology has changed a little bit. And for those of you who consume this via video, looks like a bit of a different place. I am recording from a different place here. As you'll see throughout, multiple different places. I was traveling to this place as I introduced the topic for today. I was traveling here, and I was connecting through an airport, and it was a bit of a long day, and I was looking to get something to eat, and I went by a hamburger place with quite a line, and I'm deciding, do I want to go there? Do I want to wait in the line? And I decide, no, I don't want that choice. And because I believe at one of the places next door with a really short line that there's a really good chicken bowl. By the way, a bit of fan of kava, if you've had kava, so I think that's what I'm getting. And so I say no to the burger place. I go to get my chicken bowl. And so I get this, and it turns out it wasn't at all what I was expecting. And I got something very different than that. And it turned out to be almost kind of a chicken salad type thing. And it was interesting because then I got that, and I'm walking by the burger place, and I really wish I would have gotten the burger in this setting. It's the trials and tribulations of travel. Well, it introduces a bit of my topic today, and the title of today's discussion is Regulators, False Choices, and ICH-E20. And so I'm going to try to tie this all together in what I mean by this. So I'll start by talking about the ICH-E20. Kurt and I did a discussion of this. You can go back and previous episode of the ICH-E20. It's a draft guidance document that discusses adaptive trials. Now, most of this is good. Most of the ICH-E20 is good in the sense that the existence of it, that adaptive designs are confirmatory trials. And by the way, backing up, ICH-E20 is very much a harmonization. And so what goes in there is largely harmonized, essentially agreed throughout many, many regulators, global regulators. And so in some level, this is the floor of things. Now, different regions are going to have different behaviors on things, but to some extent, a floor level thing. So from that perspective, adaptive designs are confirmatory. Sample size adaptations are confirmatory. Enrichment trials are confirmatory. Seamless trials are confirmatory. Response adaptive allocation is confirmatory. And it has a small set on Bayesian being confirmatory. All of these things are in there. Now, as you can go back to that, part of this is it's written from a pretty negative viewpoint. I'll say that, from the perspective of a lot of it is very cautionary. It's not inviting in that perspective, but yet its existence is still a positive thing. I want to jump into what I'll call the paragraph. And it's because this paragraph has been used, almost weaponized. Now, I'm going to go into this. I'm going to talk about regulators, and I do want to say this up front. I think regulators do an amazing job. And their perspective is different. I've never been a regulator. I've been proposing designs to regulators for more than 25 years, and I think they do a tremendous job. So part of this may border on what you consider criticism, and I'm going to try to put myself in the mind of a regulator and think of that perspective of what happens when you're on that side and evaluating designs. But from that side and perspective, there's a particular paragraph in ICH-E20 that I think has been weaponized, and I think the paragraph is wrong. So I thought that this would make a good podcast to be a bit controversial here. So first, in ICH-E20, there's a section that talks about, this is section 3, and it talks about the adequacy within the development program, section 3.1. And this is not the paragraph yet, but it has the sentence that says, a stepwise program with careful analysis and evaluation of completed exploratory trials helps inform the goals and design choices for subsequent confirmatory trials and ultimately generate data necessary for regulatory decision-making. Somewhat of an innocuous sentence, but it lays out this notion that a stepwise program, to some extent, is a bit of the gold standard. And that sentence isn't a problem, but it sort of sets it out that almost what we're doing now is kind of right, and things that are different than that are pushing against that. But that moves into, in the same section, and this is line 101 in this draft guidance. So this is the paragraph now. And the start of this says, the number and complexity of adaptations at the confirmatory stage should generally be limited. So that's the first sentence, and I'm going to push back on that. And then it continues, increasing either of them, so the number and complexity, increasing either of them as a replacement for a sequence of multiple trials can impair the ability to answer important clinical questions and limit the opportunity to carefully reflect on prior results to design a development program most effectively. So that's the paragraph. It does continue on. I think those two sentences are what we'll spend a good bit of this episode talking about. It says, before planning a confirmatory trial with multiple adaptations, sponsors should discuss whether additional exploratory trials are necessary to investigate the questions addressed by the proposed adaptations. So this paragraph has been somewhat weaponized in a way, and I know that sounds pejorative, but that this idea that the number of adaptations, the number of interims, and the complexity is bad. And it's a replacement for a sequence of multiple trials. Now, remember, going back, to some extent, that's the right answer here. So a couple things about this. And so, by the way, this has been presented where designs have been presented to regulators. They've come back and they've pushed back on the design, some form of no, and they've cited this draft paragraph for the reason for that. It's too complex. Do a separate trial and then a simple phase 3 confirmatory trial. So within the guidance document on adaptive trials that says all of this is confirmatory, there's a paragraph that really limits much of it to something that I'm going to push back on, and I think it's largely wrong. So first, there's this general thought that more adaptations is somehow bad or somehow not good. I think this is largely a myth. I don't believe it creates more bias. Yeah, it's operationally more complex. By the way, marginally, it's to do, 7 interims compared to 6 is marginally very little. To do 2 compared to 1 is marginally very little. That first one in setting up the process of how this is done is an operational lift. It's minor relative to the general operational burden of running a clinical trial. But I think this is largely somewhat of a myth that multiple adaptations in some way increases potential bias. And to some extent, I think it almost decreases it. When you do a single adaptation in a trial, a single futility or a single success analysis, and much of these, what I'll call myths, come from the traditional thing of the only adaptations were stopping for success, yes or no. And now trials have moved on greatly, as many of those that I read that are in this guidance, the possibility of those. So when you do one of these analyses and you continue on or you don't stop or you jump futility, there's something learned about what's going on in the trial. It's, depending on when, the relatively minor, but people can perceive there to be potential biases of that. When you do one, that's the biggest potential there. If you did 100 in a trial, each one of these, it's like daily, these are being done, essentially ignored. And we've done trials. ICH-E20 used to do interims daily. It eventually went to weekly. And I think, by the way, trials are more likely to go that way. It essentially removes these types of concerns. So I want to push back on that, but that's not even necessarily the large extent of it. It's that overall, this sets up, and this is where I'm going to get into this idea of sort of false choices. This somewhat sets up the notion that adaptations are shortcuts. That when the alternative is doing multiple exploratory trials, that somehow adaptations are always shortcuts. And by the way, when you read this ICH-E20, that's an overriding thought about this guidance document. It's the part that when you read it, much of it is set up in that way. And I think it's a false choice. And I think false choices set that up. And so I want to walk through this a little bit. I'll walk through it with examples and try to explain what I mean by that from it. So I'll start with an example trial. And this trial was conducted several years ago. The trial's over. The name of the trial is SEPSIS-ACT. And it was a trial run by Ferring Pharmaceuticals. It was the treatment for sepsis. And they had a treatment, a form of a long-acting vasopressor. And they had 3 doses and to explore. And they wanted to explore the effective dose, and they wanted to explore the effective dose on clinical outcomes, not necessarily biomarkers. So the role is a long-acting vasopressor going to be beneficial on clinical outcomes for patients with sepsis. So they have 3 doses and placebo, and the question is, what kind of trial do they run? And they want to explore dose, and then they want to run a phase 3 trial. Well, when we started looking at this, initially they thought of, okay, maybe we do a phase 2 trial, separate sequential trials, and explore 3 doses and placebo. Well, to get some idea of the effective dose on clinical outcomes, like length of organ support and mortality, the endpoint in this trial ends up being a composite of mortality and length of organ support for those that survive, an ordinal endpoint. that 200 patients or 100 patients or 200 patients was insufficient to select dose. It's too small. As you start to get into 400, 500, 600 patient trials, you're almost starting to get into phase 3 territory. So to run a realistic phase 2 trial to understand clinical outcomes starts to get to 500, 600, 700 patients. Well, now you're getting into mini phase 3 trials. So they explored this and they ended up with, I'll describe the SEPSIS-ACT trial, where when 100 patients are enrolled, equal randomization to 3 doses and placebo, response adaptive randomization starts only on the active doses. The randomization to control is fixed. And by the way, it's fixed at a third, so 33%, and then the remaining 67 are set up. So it wasn't equally to all of them, but the doses are equal and placebo's 33%. The response adaptive over these 3 doses. It also had the possibility of adding a 4th dose if the dose response looked increasing within that. So a neat little design that at 100, these RAR updates start. Once 200 patients are enrolled, and by the way, those happen monthly. So every month RAR is reset. So now we're starting to do a good number of interims in this stage 1 of the trial. Once 200 patients are enrolled, the trial could go to stage 2. That would be selecting a single dose and moving to 1-to-1 randomization, and the final sample size would be 1,800 patients, including stage 1. And that would form a phase 3 trial. And interims keep happening monthly, and it could go from 200 anywhere up to 800 could be the dose ranging portion of this trial. At 800, there's a forced decision. Now you have to go to stage 2 or you have to stop, and it's a large phase 2 trial, but largely you'd be stopping for no-go. I won't call it futility, but it's a no-go to phase 3, and it becomes a large phase 2 trial. If things are going very well and you've identified a dose, it could go earlier, and it's 1,800 is the final sample size. And so there's no adaptive sample size if this goes to phase 3. The trial ended up running more than 20 interim analyses during the course of it, and this is considered a complex design, multiple interims, and so now I'll claim that if this design would be submitted in the presence of that paragraph, it would be very easy for a regulator to look at that design and say, that's too complex, too many interims, go run a separate phase 2. Sequential trials are better as set up. Go run an 1,800 patient phase 2 trial. Then when you've selected dose, made your decisions, go run an 1,800 patient phase 3 trial. So as a regulator, maybe that's what you're evaluating. So when presented this seamless 2-3 trial with a good number of interims in the trial, a fair amount of complexity, and again, I'll back up and think that these interims aren't causing more bias. They're not somehow risking this. It's not a bad thing that these are done. I think in some ways they de-risk all of it, but when saying, oh, no, you can't do that trial, they think the alternative is an 800 patient phase 2 followed by an 1,800 patient phase 3. The problem is that's not the alternative. That's a false choice. As this extreme sort of value is, oh no, I don't want that. I'm going to get this other thing, but you're not getting that. So you say no to design A, and now you've got a paragraph that somewhat weaponizes a reason to say no to that, hoping you're going to get design B, 800 plus 1800, but you don't get B, you get design C. What do you get in that circumstance? Well, you either get that they don't explore dose and they go to phase 3, or a small 200 patient trial that actually does a really poor job at dose finding, go, no go, followed by 1800 in that. So the design you want, which is 2,600 patients in the separate, is never going to be run. It's a false choice, and the reason for that. Now, coming back to this idea within this trial is this adaptive design a shortcut? And it is a shortcut when evaluated compared to 800 and then 1800. It's a shortcut in that setting. It's combining the 800 with the 1800. Now, we can control type 1 error. We can understand all of the characteristics of this. We can make it a confirmatory trial. But it is using 1,800 patients rather than 2,600. It's efficient within that perspective. So, but compared to 800 and 1,800, you could claim it's a shortcut. But that's a design that's never going to be run. What would be run is 200 plus 1,800. And the 1,800 with the seamless up to 800 is a better design. It's a better design for regulators. It's a better design for patients. It's a better design for sponsors. Everything about it is better. And that's the value of adaptive designs here. And efficiency isn't shortcut. Efficiency is the ability to answer questions that aren't going to be done separately. Now, go back to episode 17, by the way, where I talk about a paper with Lindsay Berry, Joe Mary, and Kurt Veely. If you have 1,800 patients to use as a sponsor, and you have 3 doses to explore, a seamless 2-3 trial is going to dominate in terms of getting the right dose, in terms of power, any other sequential use of those 1,800 patients. You could break it up into 500 and 1,300 compared to a seamless trial, and that paper shows, Berry et al. Shows that that's the optimal way. Improves power, dose selection, everything about it is better than separate trials. So that's the part of this that I think's a false choice is, no, I don't want A, I want B, but you don't get B, you get C. It happened to me traveling yesterday. I said no to A, thinking I was going to get, I said no to the hamburger, thinking I was getting something else. What I got was not that. I would have gotten the hamburger had I had the choice. The same thing from the regulator perspective here. Now, what does this mean? Interestingly, by the way, that trial design was given a SPA, a special protocol assessment by the FDA. Was approved. They said yes. There were various interactions, and I think the interactions really were in what would be done. And it was presented, and by the way, this did go to multiple regulators as, why don't you do a 200-patient phase 2 trial and then a phase 3 trial? And we were able to present the different operating characteristics of this. And I think it was with the ability to see the range of potential possibilities here. This is actually a pretty good design for all parties involved, and it's not a shortcut. Adaptive designs are not shortcuts. Efficiency is not shortcuts. Depending on what you think the possibility of those designs are. Okay, interestingly, in that trial where these interims were run, I was actually unblinded during the course of this trial. Had this trial been a 200-patient phase 2, they would have run phase 3. They would have run an 1,800-patient phase 3 trial. The data at that time were quite positive. Now, by the time 800 rolled around, it wasn't positive and it stopped for no-go. And you can look up the various characteristics of the results have been reported on this. And the trial was a no-go. It didn't go to phase 3. It ended up using approximately 800 patients with it. I think the result of it was quite good. 200 patients, it would have conducted a very large, expensive phase 3 trial with CMC and the expansion to it, which I think would have been the wrong choice. Now, that's one particular outcome of the trial, but we simulated all the possibilities, and we know the probability of these things happening from a 200-patient trial is quite different from the 800, and that was part of how this trial was built. So this idea is a lot of times a design is presented to regulators, and they're evaluating that single design. And so when you're evaluating that, it's always, well, compared to what? And that's what, as somebody who for 25 plus years have been submitting design to regulators, a lot of times we submit that single design. And yes, there's a line I use quite frequently. I'm sure I've used it on other podcasts, is when somebody asks me as a statistician, hey, how's your wife? The answer is, well, compared to what? Now, my wife, by the way, listens to all these podcasts, and she's a saint, as you can imagine, putting up with this. But that's the same sort of thing. It's, well, compared to what? And if you're in the mindset of, okay, I've been presented this design A, boy, I'd rather have them do separate trials, big trials sort of thing. If I'm thinking that's my comparison, I might say no to A because I think I'm getting B. But B is not possible and it's not going to be run, and you get C. So a recent example of this, so part of this is how do we help this process? And if I were a regulator, I might very well do the same thing. So I don't want this to be a criticism of regulators. They sit in a different chair and they're evaluating a single design, a single thing. How is that? Well, I mean, compared to what? And I might think, oh, you should do this adaptive design sort of thing. So I push back on A, but I don't get a better adaptive design. Maybe I get a design I think is worse than that one. So that's a challenge in this interaction. You usually throw a single design and say, hey, how about this design? A recent example of this was another seamless 2-3 trial. It explored 2 doses and a relatively rare disease, by the way, exploring 2 doses and picking a dose, moving to 1-to-1 randomization, and going to a section that is then adaptive sample size, so 3 interims to select sample size once it moves to 1-to-1. And that's the trial design and submitted to regulators. Now, there's some pushback on that. And again, that paragraph could be used to say, aha, you're doing dose selection, you're doing several interims. That's too complex to be confirmatory. Go run a separate phase 2 and then run phase 3. And this was actually the advice given by regulators, given by the FDA within that. When this comes back and you're working with the sponsor and you say, okay, how about the possibility? Oh, and by the way, part of the reason was the pushback is the endpoint in this trial is a relatively long-term endpoint. And so we're making dose selection based on early predictive markers. Now, those markers are not the confirmatory marker endpoint at the end, which is the longer-term agreed feels, function, survives endpoint. But we have to, because of enrollment and because of the scenario, base it. There will be a little bit of long-term endpoint, but largely it's an earlier endpoint. And the FDA was concerned about the predictability of that, and they would rather have a separate phase 2 that goes all the way out, long-term clinical outcome, make your decision and run phase 3. And that was the advice given. So now the regulator gets this design A and thinks, oh, that would be a better design. And you can always create a superset design to an adaptive design that is better. But that would then be this larger, longer phase 2, larger phase 3. And when going back to the sponsor and saying, okay, is that a possibility? It's impossible. They said the company would fold. That trial can't be run. So that's where this falls into it, is any adaptive design, you can create a design that's a superset of it and say, oh, you should do that. It's not a possible choice. You can't do that. So saying no to A because I want B is a false choice. So if regulator says, no, you can't do design A, what gets run is a phase 3 trial without dose selection. And that's what the sponsor said. If we can't run that trial that selects a dose based on earlier interim, we're just going to pick a dose and run phase 3. What you get back is not why you said no to A. So all of a sudden, suppose the regulators understood that the choice was that design A that at least selects dose based on an earlier marker, and then moves into the seamless phase 2-3 part of it, into the confirmatory part, or you just get a phase 3 and they pick the dose with no data. I think they'd pick design A, and they'd live with picking it on that endpoint. But when evaluating it in their mind, they're thinking, oh, I think you should do design B, but that's not a choice. Now, this is a bit of a challenge for us, is how do we work with that? How do we present this in a way that's consumable and all of that? And I'll come back to that a little bit. What happened here, by the way, is a great story, is in the in-person meeting, the sponsor shared that we can't run that design. We can't run separate trials. It would not be run. The agency became a partner in this sort of development as, where do we go? What can we do under this setting? And largely a form of A, slightly adapted in a good way. It was what came out of that. So it was a beautiful interaction. And again, I think the FDA did a tremendous job in being partners in creating better care for patients. And it was great, but it's this hard aspect of evaluating a single design in isolation. Another example of this, and I'll do this relatively quickly, was an enrichment trial where it started off 1-to-1 randomization, phase 3 trial, but there's a concern about what's the right patient population. And there's some risk that we might be enrolling patients who don't benefit. There's always this enrolling people on the healthy side or the less healthy side. Maybe that you can't really you can't modify the effect in those patients, ceiling and floor effects. So it starts off enrolling a rather larger set, but could restrict enrollment. Do enrichment in the trial. And to do enrichment well, by the way, when you simulate these trials, you have to do it relatively early. If you wait too long to do enrichment, your trial's largely enrolled and it has little benefit in it. So you need to do it relatively early. So the trial had 3 interims for potential enrichment, and then had 3 interims for sample size. So all of a sudden, this is a 6 interim phase 3 trial. This literally referenced the paragraph that I gave you as this is too complicated, and doomed it by saying this trial, it makes, because of the complexity and the adaptations, it makes that trial exploratory. And that's death. It means it's not a confirmatory trial. By the way, a different regulator region said, yes, the design's good. We like it, which is a challenge here. But I think the regulator that said, no, this is exploratory, and used that paragraph as a weapon, thought they'd get a different design. Run that trial to understand the patient population, and then run phase 3. But that trial's never going to happen. It's unaffordable, the time. Those large phase 2s don't happen in that. What would happen is they guess at the patient population, and they don't actually explore that, and they run a phase 3 at risk. Or they ignore the region that said no to the trial, which neither one of those is a better option than trial A of what they got. And so that's a huge aspect here, and such a challenge in these circumstances of being a regulator, because maybe you don't know what the alternatives are. If you reject the null, what's the alternative? If you reject a design, what am I going to get? And maybe I would go back and actually really like the one that was already submitted. And so several parts of this, again, that design, this, call it 1,000 patients in this enrichment phase 3 trial. What they'd rather have is 1,000 patient exploratory trial to find the enrichment, or the enrichments only can happen up through 400, run a separate 400, and then run 1,000 patient phase 3. But that's never going to be run. That trial is more efficient, not more efficient, but more powerful than this seamless 1,000 patient trial. But that's not a choice. And so this now does better than all the alternatives, and the adaptations allow a better trial to be run for everybody. Another very, very simple example is a very common example. This isn't a real one, but it's reflective. The other ones were real examples. Is reflective of what happens quite a bit is a sponsor is interested in running a phase 3 trial, 1-to-1 randomized, double-blind, all the nice things. And they say, okay, for an effect that they think their drug has of delta, they get an 80 or 90, it doesn't matter, percent powered trials, 180 patients. And then you show them, well, a smaller effect than delta, which is still clinically meaningful, might only have 50 or 60% power with your 180. But they don't want to run a 300 patient trial because they think they're better than that, and they think that's a waste of resources. So if given the choice of a single fixed trial, they're going to run a 180 patient trial. Now, the risk is that there's a clinically meaningful drug that doesn't hit success with that. So you show them, what if we do several adaptations for sample size? And suppose we do interims at 150. So if the drug's even more effective, we get out sooner. 200, 250, or 300 with appropriate futility rules and success looks. And all of a sudden, this design performs really, really well, because if their delta is true, they still get out with something that on average is 180 or better than that. They stop at 150, 200 with high probability. And that's really good. If it's clinically meaningful but less than that delta, they still have a good chance of success, or 250 or 300. So a really nice design. Now, if somebody says that's too complex, too many interims, they might think that because they're getting a 300 patient trial. I say no to that, because run the 300 patient trial, but the 300 doesn't get run, the 180 patient trial gets run. So that's the challenge of this. This is the challenge of a regulator, and this is not necessarily their fault in thinking about what this alternative space. And if I were a regulator, I might have a different alternative set of designs that I think could be run. And so I say, oh, you should do the following, but those are never going to be run potentially. And I get something back as a reaction to this that's worse than the original one. And these are the challenges of the way this is particularly done. So I think we need to communicate that better, which is hard, by the way, because you can't say, if you don't take A, we're going to do C, in a way that is almost blackmailish, that, boy, if you don't say yes to our design, you're going to get this. And that's really not the way. We traditionally, when we submit a design like that, we work really hard to explain that single design as to what you're going to get as a regulator. Now, we don't spend a lot of time comparing to a bunch of other designs. It's actually can can be confusing to somebody receiving this. What's the design again? But I think we need to do a better job of that to understand the breadth of the designs that could be run, not in an aggressive way, and showing that this is a better design. A seamless 2-3 trial design's better design than a lot of the alternatives that could be run in that scenario. And so this is really our challenge as clinical trialists, to be able to do this in this weird interaction we have with regulators. Tied to it is, as this draft guidance on ICH-E20 is going, hoping a bit of that paragraph can be massaged and less of a sharp weapon for moving forward in clinical trials. All right. I appreciate you joining me today in my new digs. And yes, I survived the travel and in all of that. And so I appreciate you joining us. Until next time, we'll be here in the interim.